Welcome to this summary on the cellular mechanisms underlying myelin changes and clearance in aging optic nerves. This video explores recent discoveries regarding myelin outfoldings, myelinoid bodies, and their interactions with glial cells in mouse and human nervous systems. Myelin, a multi-layered sheath produced by oligodendrocytes, is essential for rapid nerve impulse conduction and axonal health. As organisms age, myelin integrity declines, contributing to cognitive and sensory impairments. The question addressed here is, how does aging affect the ultrastructure of myelin in optic nerves, and what are the mechanisms by which myelin debris is cleared? Understanding this is crucial because disrupted myelin homeostasis is implicated in neurodegenerative diseases and age-related neural dysfunction. The approach uses advanced electron microscopy combined with immunolabeling and organotypic slice cultures. By analyzing diverse ages in mice and comparing to human tissues, the authors investigate changes in axon calibers, myelin sheath morphology, formation of myelinoid bodies, and their interactions with astrocytes and microglia. Moreover, they explore the physical processes by which these glial cells remove myelin debris, proposing a model of trogocytosis involving stretching and tethering of myelin remnants. This integrative approach, combining ultrastructural, immunological, and dynamic live imaging techniques, provides insights into age-associated myelin turnover and clearance mechanisms. Figure 1 illustrates the age-related ultrastructural changes in mouse and human optic nerves. Panel A shows transmission electron microscopy cross-sections of chemically fixed optic nerves from mice at 6 months and 24 months. In these images, myelin debris, termed myelinoid bodies or MBs, are marked with asterisks, and myelin outfoldings are indicated by arrows. Quantitative analysis in Panel B reveals that older mice exhibit a significant shift toward larger axon calibers, accompanied by an increased number of myelin lamellae. Small diameter axons showed decreased frequency with age, while axons larger than 0.8 micrometers maintained normal myelination with additional layers matching increased axonal calibers. Moreover, myelin outfoldings often coil around neighboring axons and become more numerous and larger with age. Importantly, MBs also increase in both number and size as mice age. The authors observed that on electron microscopy, MBs frequently reside near, but are not physically connected to adjacent myelin sheaths, suggesting they originate from myelin but are separable entities. Similar patterns of MBs and outfoldings were observed in human optic nerves, though the MBs tend to be smaller. This figure thus depicts how aging is associated with progressive myelin morphological abnormalities, including the formation of outfoldings and myelin debris accumulation in the optic nerve, illuminating key structural hallmarks relevant for age-related neural decline. Figure 2 investigates the cellular localization of myelinoid bodies, or MBs, in aged optic nerves using colloidal immunogold electron microscopy with markers for myelin proteolipid protein, PLP, and glial fibrillary acidic protein, GFAP. Panels A through C demonstrate that MBs localize extracellularly and intracellularly. Specifically, MBs were detected within a non-compact outer tongue region of the myelin sheath around axons, within microglia, and within astrocytes, identified by their characteristic markers and ultrastructural features. Microglia showed membrane stacks and cholesterol crystals that likely indicate advanced myelin degradation stages. Astrocytes contained PLP-positive lysosomes, supporting their active role in degrading myelin debris. Panel D shows high-pressure frozen electron microscopy images where extracellular MBs of varying sizes appear closely associated with both microglia and astrocytes. Panel E quantifies the density of tiny MBs, defined as having fewer than 10 lamellae and diameter under 300 nanometers, showing these are predominantly extracellular and increase with aging. These tiny MBs include both those with electron-lucent centers and membranous centers, indicative of distinct structural configurations. Statistical analysis confirms the age-associated increase. Collectively, the authors interpret these data as revealing that MBs are found both inside glial phagocytes and in the extracellular space, suggesting complex dynamics of myelin debris clearance involving multiple glial cell types in aging optic nerves. In Figure 3, The authors utilize focused ion beam scanning electron microscopy, FIB-SEM, for in-depth three-dimensional reconstruction of myelin sheaths, outfoldings, and associated myelin debris in the aged mouse optic nerve. Panel A presents 3D reconstructions showing myelin outfoldings appearing as finger-like sheets, extending on average 9 micrometers in length. These outfoldings contain a continuous cytoplasmic compartment connected to the inner tongue of the myelin sheath. Notably, 2 myelinoid bodies are shown connected to a myelin sheath and an outfolding respectively by thin myelin tethers. Panel B classifies and quantifies extracellular and intracellular MBs within astrocytes and microglia, based on 1,200 consecutive EM images. Surprisingly, only a minority of MBs previously thought to be isolated in 2D images are truly detached. Most remain tethered to myelin sheaths or are misclassified due to distorted morphology. These tethered MBs, or TMBs, constitute the largest category both inside and outside phagocytes. Panels C through F provide reconstructions revealing that TMBs are physically linked to myelin sheaths or outfoldings via thin tethers within microglial or astroglial processes. These tethers often show subtle distortions, consistent with mechanical forces applied during engulfment. The authors interpret this data as indicating that most MBs are not free debris, but remain structurally connected to intact myelin sheaths, suggesting that glial-mediated clearance of myelin involves the pulling and severing of these tethered structures, rather than engulfment of entirely free debris. Figure 4 focuses on the structural properties and fate of myelin tethers connected to myelin sheaths and myelinoid bodies using FIB-SEM analyses and immunogold labeling. Panels A through C present 3D reconstructions of mouse optic nerve tissue from a 20-month-old mouse, showing an astrocyte engulfing an MB with a short tether, not connected to a myelin sheath, a thin blind-ending tether linked to a myelin sheath by a finger-like protrusion, and a tethered MB engulfed by astrocyte with a tether connected to the tip of a myelin outfolding. The presence of bending membrane profiles supports active membrane fusion and fission processes within the tether. Panel D shows 2D immunogold labeling for proteolipin protein, indicating that tethers have fewer myelin layers than the associated MBs, and lysosomes containing myelin protein-positive material are evident in astrocytes, highlighting ongoing degradation. Importantly, panels E and F demonstrate that similar thin myelin tethers are also detected in aged human optic nerves via Luxol fast blue staining and scanning electron microscopy. These tethers associate with myelin sheaths, dystrophic myelin or myelinoid bodies, and are morphologically distinct from outfoldings. The authors interpret these observations as supporting the concept that myelin tethers become thinner and eventually rupture, releasing MBs that are subsequently cleared by glial cells, representing a key step in age-related myelin turnover conserved across species. Figure 5 examines the presence of myelin debris within glial cells in the aging mouse brain, focusing on the hippocampal stratum lacunosum molecular region. Panel A shows representative immunofluorescence images confirming the presence of myelin basic protein, MBP-positive debris, within GFAP-labeled astrocytes and IBA1-labeled microglia in 24-month-old mice. Panel B presents 3D confocal projections of individual astrocytes and microglia demonstrating MBP inclusions, indicating internalization of myelin debris. Panel C quantifies the volume of myelin debris contained within microglia and astrocytes, and the percentage of these cells exhibiting myelin debris at 6 and 24 months. Results reveal that both astrocytes and microglia show increased myelin debris uptake with aging, with microglia containing a somewhat larger volume. There is also a greater percentage of microglia and astrocytes containing debris in aged mice compared to young. These data suggest that both microglia and astrocytes contribute to clearing myelin debris in the aging brain, consistent with observations in optic nerve tissues. The authors interpret these findings as demonstrating active roles for these glia in maintaining myelin homeostasis during aging, through phagocytic clearance of damaged myelin. In Figure 6, the authors use cortical organotypic slice cultures to visualize and analyze microglial interactions with tethered myelinoid bodies, or TMBs, in a controlled environment. Panel A presents confocal microscopy images at 21 days in vitro, demonstrating robust myelination with fluorescently tagged myelin and microglia markers. Panel B zooms in on a single oligodendrocyte expressing membrane-tagged EGFP, showing compact myelin ultrastructure. Panel C provides corresponding transmission electron microscopy images, validating the layered compact and non-compact myelin sheathing axons. Panel D displays time-lapse confocal sequences tracking microglial processes associated with TMBs connected to long myelin internodes. Microglia dynamically extend and retract processes to pull on TMBs, resulting in stretches averaging 0.59 micrometers per minute and maximum stretches up to 44 micrometers. Panel E quantifies the velocity of these tether stretching events, while Panel F analyzes the directionality of TMB movement relative to microglial positioning. Panel G shows electrophysiological patch clamp recordings, indicating increased resistance consistent with myelin sealing and tether pulling. Panel H captures 2-photon microscopy images, documenting TMBs pulled with a patch pipette, showing elastic myelin tethers stretching over 200 micrometers before rupturing. Panel I offers a 3D rendered visualization of a pulled TMB. These results support a model in which microglia actively exert physical forces to stretch and sever myelin tethers for debris removal, highlighting a dynamic mechanistic component of myelin clearance involving microglial trogocytosis in living tissue. The final figure, Figure 7, proposes a working model explaining how myelin debris removal occurs through a trogocytosis-like mechanism executed by microglia and astrocytes. The schematic depicts that trogocytosis involves 2 key steps. First, stretching of tethered myelin bodies applies lateral tension specifically at the outer edges of the pulled myelin tether. Second, this mechanical tension forces ordered consecutive delamination at the intraperiod line, a 4 nanometer wide interface between myelin lamellae, along with intermembrane fusion and fission events of the innermost tether lamellae. This model accounts for the observation that after removal of tethered myelin bodies from an internode, the residual myelin sheaths remain structurally organized with intact multiple compact layers. The authors interpret these fusion profiles, membrane bending, and loop-like membrane structures observed ultrastructurally as signatures of the trogocytosis mechanism. This conceptual framework integrates morphological, biochemical, and biophysical data into a coherent mechanism for selective and progressive clearance of myelin debris by glial cells, maintaining myelin integrity, while allowing aged or damaged myelin to be removed efficiently. In summary, this body of work elucidates how myelin undergoes structural changes with aging, including increased axon calibers, myelin outfoldings, and accumulation of myelinoid bodies. The key insight is that these myelin debris structures largely remain tethered to intact myelin sheaths and are actively cleared by astrocytes and microglia. Through advanced 3D electron microscopy, the authors reveal that the common notion of freely floating myelin debris is incomplete. Rather, the glial clearance process involves stretching and severing of thin myelin tethers. Live imaging in cortical slice cultures shows microglia dynamically pulling on these tethers, exerting mechanical forces that ultimately lead to tether rupture and debris engulfment. The proposed trogocytosis model explains how the myelin sheath retains its spiral organization after partial removal of tethered myelin bodies, highlighting a highly regulated mechanism of myelin turnover. This study advances our understanding of glial myelin clearance and aging, and sets the stage for exploring therapeutic approaches targeting myelin maintenance in neurodegenerative diseases and age-related cognitive decline.